Okay, Bismillahirrahmanirrahim. Assalamualaikum and good morning to everyone. So this is the second part of our chapter 3 for plant physiology on the plant growth and development. We should have covered this part of chapter 3 last week, but due to sickness, I couldn't continue it. So for today's, for this week's punya lesson, we're going to do it on FOC. Why I give you today? Because this part of the lesson, we're going to do a practical on it on the same day. So I really hope you can listen to my lecture, and then in class, if you have any problems, if you have any questions, you can come right up to me, and hopefully we can do the practical, the labs smoothly. Okay, so the second part of chapter 3 on plant growth and development, we're going to have a look at the major groups of plant hormones, or other people, they prefer to call it plant growth regulators other than hormones, because these chemical compounds that is naturally occurring in the plants, it regulates the plant growth and development. So that's why they prefer more to call it the plant growth regulator rather than plant hormone. So as mentioned just now, hormones they are naturally occurring, so they are organic because naturally available from plants, and even at low concentration it can have an obvious influence in the physiological processes of the plants. So for the plant hormones, the site of synthesis is diffuse, and the response of those plant hormones or the plant growth regulators, it can depend on the sensitivity of the target cells. And also we have to note that even though we have several groups of plant hormones, but several hormones, they can have one effect only. So for this part, there are two objectives that we are trying to reach. The first one, to be able to determine the effect of various hormones in plant growth, and the second one is to be able to monitor the differences in plant responses. The first major group of the plant hormone is the auxin, and we're going to have a look at what auxin is, who found auxin, and we can see how to create a simple experiment on to investigate the effect of auxin on plants. So auxin, in other words, is the IAA, or it stands for the indole 3-acetic acid, and these are the chemical structure for it. So auxin is first described by the Fritz when and isolated by the Kenneth Lehman, and auxin, it has numerous roles in plant activities, which is listed in front of you here, but usually auxin is popular and famous for their root initiation and development. If I can try do this. Okay, tangan menggeletar. Okay, I better stop now. Alright. So auxin, as I mentioned just now, dia memang famous and popular for the root elongation because it stimulates adventitious root growth. Besides the adventitious root, it can also originate from the callus cells. So that's why if we grow the stem cuttings, if we grow the plant from the stem cuttings, we can also have root from it, due to the production of auxin from the plants. However, if the high concentration of auxin available in plants, it is not stimulating the root anymore. It is now inhibiting the elongation of the root. Besides auxin, we have to know two things. It has effect on the root, and it also has effect on the shoot or the bud formation. in terms of the root, auxin promotes root development, initiation, and elongation. But for shoot or bud formation, it inhibits. So auxin doesn't like bud formation, but auxin loves roots elongation. So this is a simple experiment on how to see the effect of IAA or auxin or indole 3-acetic acid to the root formation. So you can do this simple experiment at your home, in fact. So we can try using the coleus. This is coleus plant. This is native to Malaysia. I believe you must have seen this around your places, the gardens, ataupun your back home lah kan. So in Melayu we call it pokok hati-hati atau pokok hati-hati. Some people call it pokok hati-hati, like careful, hati-hati tu, ataupun pokok hati-hati. So these are the simple methodology, simple experiment to show there is an effect of auxin or the IAA on the root formation. And also, I mentioned just now, we have two effects of auxin on plants, which is one on the root and the other one on the bud formation. So this is the simple experiment on the bud formation as well, and this is the results for the root formation. And we can see here a lot of roots, but here no roots at all, or maybe very minimum. Okay. So we can see that auxin or the IAA or the indole 3-acetic acid, it promotes root elongation and initiation. And this is on the bud formation, and we can see there is a bud coming out here, but here there is no bud formed. That's the first major group of plant hormone, which is the auxin. And we have to remember again, auxin, it is popular for root elongation, root initiation, but it doesn't like, it inhibits the formation of bud. Okay. Now, the second group of the plant hormone or the plant growth regulator is the gibberellin. Gibberellin or the gibberellic acid, or short form is GA, it is discovered by the Kurosawa through the fungus in the genus of Gibberella. So we can see here kan, the name of the gibberellin or the GA tu, it comes from the fungus. And it's the same with auxin. Gibberellic acid ni, they have a lot of effect on the physiological roles in plant, but mainly on the stem. Okay, of course it can also promote the seed germination, the carbohydrate mobilization, in the internode elongation, but gibberellin ataupun GA ni is famous for stem stimulation. So the site of synthesis of this GA ataupun gibberellin is on the developing seeds, developing fruits, young leaves, apical region of roots. But we have to remember that the GA, it does not stimulate flowering. And also we can see here, GA, it induces early production of seeds by some biennials after only one season instead of two. So we know that biennials, it means that there are two seasons dekat situ, But when we have a lot of GA ataupun gibberellin, then it can cut short dia punya production from 2 seasons to 1 season only. So this is a simple experiment to investigate the stem growth with the effect of gibberellin. Okay, so I believe this is the same picture, tapi it shows a different part. Tadi it shows dia punya root, tapi now it shows the internodes growth tu for gibberellin. Okay, so recap for gibberellin. Gibberellin, or the other name is the gibberellic acid, ataupun dia punya short form dia GA. They are popular and famous for stem elongation. Okay. Oops. Okay. Now cytokinin. So cytokinin ni first described by Johannes van Overbeek, and same as other groups, they also have important roles in plant, but mainly it is for delaying leaf senescence. So cytokinin works in tandem with auxin to cause cell morphogenesis and division. So dia promote lagi cell to morph, and also to divide. So that's why it delays the leaf senescence. So if you can see here, if they have exogenous supply of cytokinin, it means that we add more cytokinin. We do not depend only on the natural cytokinin, then the plant senescence can be delayed. And another fun fact is, the coconut water has the hormone of cytokinin, which the seed uses when it germinates. Okay, because just now kita boleh nampak one of their physiological roles of cytokinin is on the seed germination. And a new fact that we learned today, that coconut water, it has the cytokinin in it, which can promote seed germination. So for that, a simple experiment. They are using 3 different petri dish. The first petri dish, they just add distilled water. The second petri dish is on the coconut water, and the third one on the pure cytokinin solution. This is to check on the growth and also to extract their chlorophylls. Unfortunately, there is no result on it. I couldn't find the result, but we can. It makes sense that the petri dish with the cytokinin and the petri dish with the coconut water will germinate the seeds and also will delay the leaf senescence. Okay, and the last major group of the plant hormone is the ethylene. And this experiment is the simplest experiment ever to investigate the ethylene effect on the plant growth and development. So the source of ethylene, it comes from the ripening fruit, because as it ripes, it will cause changes to the fruits. And what kind of changes? The breakdown of the chlorophyll, synthesis of other pigments, and there is a softening due to the cellulase and pectinase, those 2 enzymes, and it converts the starch acids to sugars, and also the disappearance of phenolics. Excuse me, sorry. Okay, sorry, malas nak cut. So ethylene, it stimulates the female flowering expression, and ethylene, it enhances the rate of senescence. In contrast to the tadi yang kita belajar on cytokinin, it delays leaf senescence, tetapi for ethylene, it enhances the rate of senescence. Okay, and we've learned that there is a senescence and also the PCD, the programmed cell death. Okay, and aging is associated with the loss of chlorophyll as leaves fade or turn brown. Okay, and the chlorophyllase, an enzyme that its job is to break down the chlorophyll during aging and also the senescence. And ethylene can also control the abscission, leaf abscission, depending on the balance with the oxime. So this is the simplest method ever to investigate the effect of oxime on plants. So again, using the coleus plants, we put a cut apple, so any fruit actually. So if you have pear, lye or apple in your fridge, just sacrifice half of that one apple and put it with the pot. And we can see that the sample, the plant without the ethylene or without the apple, they will have more chlorophyll because no chlorophyll being degraded, and they are not in the aging or senescing mode. Okay, so in contrast to the sample B with the ethylene, with the cut apple, they have reduced chlorophyll concentration. So it shows that with the ethylene released by the cut apple just now, it promotes leaf senescence, which reduces and degrades the chlorophyll content. So in conclusion, remember we have two objectives. Okay, so we're trying to conclude our both objectives. The first objective is to be able to determine the effect of various hormones in plant growth. So this is a recap. So the first major group is the auxin or the indole triacetic acid or the IAA. And again, remember the auxin too, it has effect on both on two different parts of plants. The first one on the root and the second one on the bud formation. The second major group is the GA or the gibberellic acid or the gibberelline, and it has effect on the stem elongation. The third major group is the CK or the cytokinin, where it delays leaf senescence, in contrast to the last major group of the plant hormone, which is the ethylene, where it promotes leaf senescence. The second and last objective that we're trying to conclude is to be able to monitor the differences in plant response. So from the simple experiment that we set up just now, we can see that plants respond differently to different hormones. So it forms the roots. So this is from the auxin, inhibition of bud formation, auxin as well, elongation of the internodes, this can be from the GA, right, ataupun gibberellic acid, leaf senescence and abscission, this can be from the cytokinin and also the ethylene. Okay, it depends whether it is promoting ataupun delaying leaf senescence. So that's all for part 2 of chapter 3. I really hope you understand. If you don't understand, you can discuss among your groups, your friends, and if you have further discussion, you would like to further discuss with me tomorrow during the lab, please feel free to do so, and please use the information to do the lab practical. The lab manual, I have uploaded it into the Edmodo, and please read it carefully. And I'll see you guys tomorrow. Thank you.